Hello and welcome to my channel Sam Biology Class. Friends, today we will discuss about the binary vector. Before that, will you please consider subscribing my YouTube channel? Your subscription is often a lot to me. Wild type TI plasmids cannot be used directly for agrobacterium mediated gene transfer because these plasmids are large and therefore difficult to manipulate, manage and maintain. Moreover, with the help of wild-type plasmids, Agrobacterium tumefaciens infect only dicot rather than monocot plants. It causes Crohn gall disease in which there is an uncontrolled growth of tumors. In addition to this, wild-type plasmids do not have essential sequences such as multiple cloning site, promoter, terminator, suitable origins of replications and selectable marker genes for identification and selection of transformed bacterial and plant cells. All these problems are overcome by disarming wild-type TI plasmid. A disarmed plasmid is one from which oncogenes or tumor-inducing genes and other non-essential segments have been removed. In addition to this, other sequences such as promoter, terminator, multiple cloning site, origins of replications and selectable markers are also added to the disarmed plasmid. Disarmed TI plasmid often uses a binary system which consists of two separate plasmids. The term binary vector literally refers to the entire combination, but the plasmid which contains the gene of interest or the transgene is usually called binary vector. Out of these two plasmids, the binary vector contains only the left and right border from TI plasmid. These border sequences are recognized by Vir D1 D2 endonuclease, which together form a complex known as the relaxase complex. Vir D1 D2 proteins are responsible for precise excision at this end of the border sequences. In between left and right border, a multiple cloning site has been placed into which the gene of interest or the transgene can be inserted. In addition to this DNA segment also contains a selectable marker gene for identification of transformed plant cells. Both the gene of interest and the selectable marker are associated with specific promoter and terminator sequences. Promoter is the specific region at which RNA polymerase bind in order to start the transcription of the associated gene. On the other hand, terminators serve as a signal for termination of transcription. In addition to these regions, the binary vector contains two replication origins, one for E. coli and the other for Agrobacterium. There is also a selectable marker gene for identification of transformed bacterial cell. So by removing all the oncogenes and other non-essential segments, the size of the binary vector has become very small so that it can be easily manipulated in E. coli. The other component of the binary system is a helper TI plasmid which contains only the virulence region which is derived from TI plasmid. This Vir region contains different Vir genes which encode specific proteins which are responsible for the precise excision, transfer and integration of this DNA segment into the plant genome. This helper plasmid contains another origin of replication and other sequences. For agrobacterium-mediated gene transfer, the gene of interest is first placed in the multiple cloning site and then the resulting recombinant binary vector is rapidly amplified in E. coli. E. coli is selected because it grows rapidly and its genetic is well understood. After amplification, the recombinant vector is introduced into agrobacterium along with the helper plasmid. In the next step, transformed agrobacterial cells are identified and then used to infect the target plant. It is because of the presence of virulence region on the helper plasmid infection occurs but Crohn's gall disease do not develop. This is due to the removal of tumor-inducing genes from the binary system. During the course of infection, this DNA segment, which contains the gene of interest, is transferred into the plant cell as a single-stranded DNA molecule known as T-strand through a specialized type 4 secretion system. After transfer, the T-strand is incorporated into the plant nucleus and then integrated into the plant genome, usually at random locations by illegitimate recombination. As a result, the plant becomes genetically modified. So friends, this is all about the binary vector and the process by which agrobacterium-mediated gene transfer takes place to produce genetically modified plant. For more updates, subscribe my YouTube channel, Sam Biology Class.